Welcome back to Chapter 3, Amino Acids, Peptides, and Proteins. In this lecture, we'll be looking at Part 4 of this chapter. Here, we'll continue our discussion on peptides and proteins. We'll begin the discussion by looking at separation and characterization methods for proteins. And then we'll continue with methods to sequence proteins, and then end our discussion by looking at protein evolution. SDS electrophoresis is an important technique for the separation of proteins. This method is based on the migration of charged proteins in an electric field. This technique is not generally used to purify proteins because simple or simpler alternatives are usually available, and electrophoretic methods often adversely affect the structure and the function of proteins. However, as an analytical method, SDS Gel Electrophoresis is extremely important. One of the advantages is that proteins can be visualized as well as separated by permitting to estimate quickly the number of different proteins in a mixture or the degree of purity of a particular protein preparation. Now, SDS electrophoresis uses a polyacrylamide gel, and hence the name polyacrylamide gel electrophoresis. In addition to this, one needs to denature their protein samples so as for the proteins to migrate in a polyacrylamide gel according to their size and charge. What I mean by denatured is that the proteins has to be unstructured. Proteins in their native form have a three-dimensional structure, right? It could be a tertiary structure or a quaternary structure. When you denature a protein by either applying heat or changing pH, you destruct this tertiary structure, and as a result it becomes denatured. Now there's one more thing that is required, that is to add charge to this protein, and to do that we typically use a detergent called as SDS, or sodium dodecyl sulfate. Sodium dodecyl sulfate helps in two ways. First, it helps in denaturing the proteins because it's a detergent, and detergents are usually harmful to proteins at higher concentrations. Now, in addition to it, SDS also adds negative charges to proteins, which help them to migrate in an electric field. SDS electrophoresis is a very important technique, and in this technique, electrophoresis of proteins is carried out in gels. As I said before, it's a cross-linked polymer of polyacrylamide and these gels. Now these gels act as a molecular sieve, slowing the migration of proteins approximately in proportion to their charge to mass ratio. Migration of these proteins may also be affected by their shape. Now the way this experiment is performed is that you place this gel in a gel cassette and then add your samples to these small wells. And these, the amount of sample or the volume of sample that you could add to these wells are limited by the size of these wells. Now once you add these samples and close this cassette and apply an electric field, right? There's negative here and positive here. And SDS is treated with a protein sample, which provides negative charge to the protein, right? A protein will bind 1.4 times its weight of SDS, which is one molecule of SDS for each amino acid residue, right? This gives a net negative charge to the protein, thereby conferring a similar charge to mass ratio. Once you run the sample in this gel, right, proteins of different size will migrate differently. as you can see, the gel on the right hand side, in this case, this is after the electrophoresis is done. Once the electrophoresis is finished, you take out this gel cassette and remove the gel, and you stain this gel with a dye called Kumasi blue. And Kumasi blue is a hydrophobic molecule that binds to the proteins, thereby providing blue color, right? And that is how you can monitor these proteins. And when you compare these proteins with protein markers, right? There are standard protein markers available commercially, and they have specific sizes. And what you do is you compare your unknown proteins with their sizes. And that is how you know that your protein migrates at a specific molecular weight. Now, what are these lanes? There are like 1, 2, 3, 4, 5, 6, 7 lanes for samples and 1 lane for a marker. Right? Now, the ones 1, 2, and 3 are the crude cell lysates that we talked about in the previous part. Right? Lane 5 is from ammonium sulfate precipitation, and then after you purify by ammonium sulfate precipitation, you take it up for anion exchange. After anion exchange chromatography, you do a cation exchange chromatography. And then after cation exchange chromatography, you get pure protein. And that's what you see. And as you can see, these lanes correspond to the protein, right? And the approximate molecular weight of the protein is around 45,000 Daltons. Remember, the molecular weight of proteins are expressed in Daltons. Other separation methods include isoelectric focusing, or IEF, and two-dimensional gel electrophoresis. Proteins can be separated by their isoelectric point by applying a pH gradient in addition to an electric field. This method is known as isoelectric focusing. This method is slightly different from SDS Gel Electrophoresis in the sense that this does not use size but uses a pH gradient in addition to the charge that is being applied. Now, when a protein mixture is applied on one end of a strip, each protein in a mixture migrates until it reaches the pH that matches its pI, as you can see here. Proteins with different isoelectric points are thus distributed differently throughout the gel, and thus you can separate various different proteins in a mixture. Two-dimensional gels use a combination of size and IEF to further separate proteins. In two-dimensional gel electrophoresis, proteins are first separated by isoelectric focusing in a thin strip gel. This gel is then laid horizontally on a second slab-shaped gel, and the proteins are separated by SDS polyacrylamide gel electrophoresis. Horizontal separation reflects differences in pI. Vertical separation reflects differences in molecular weight. So horizontally, it's the differences in pI, and vertically, it's the differences in molecular weight. The original protein component is thus spread in 2 dimensions. And this is how it looks like, right? Thousands of cellular proteins can be resolved using this technique. Individual protein spots, as you can see, can be cut out of the gel and identified by a technique called mass spectrometry. This is a very useful technique when you want to study individual proteins and understand what that protein identity is using mass spectrometry or other technique that we'll talk about later. Once you purify your protein, and if your protein is an enzyme, you can use specific activity to measure your purification efficiency. Specific activity is often used as a gauge of how well your purification works, especially for enzymes. Specific activity is given as the number of enzyme units per milligram of total protein. Right? Enzyme activity divided by total mass of protein. And specific activity differs from enzyme activity. Right? Specific activity is a measure of enzyme's purity. It increases during purification of an enzyme and becomes maximal and constant when the enzyme is pure. After each purification step, you should check the activity of the preparation. Right? That determines how pure your enzyme is. Now, what is the difference between activity and specific activity? Now consider this example. There are two flasks and there are different marbles in both these flasks. All these marbles can be considered as proteins. Both these flasks contain same number of red marbles if you count them, but different numbers of marbles of other colors, as you can see. If the marbles represent proteins, both flasks contain the same activity of the protein represented by the red marble, right? If they have same number of red marbles. The second flask, however, has the highest specific activity because the red marbles represent a higher fraction of the total. This means that of all the proteins in here and here, the fraction of red marbles is higher in this flask. So the specific activity for this flask will be higher. And you can say in terms of protein purity, the red marbles in this flask has higher purity as compared to this one. Right? Now that's the difference between activity and specific activity. So specific activity is a measure of how pure your enzyme is. This table gives you an example as to how specific activity increases as you increase the purification steps of an enzyme. Right? Now, as you can see, activity decreases after each purification step because there is always some loss due to inactivation or non-ideal interactions with chromatographic materials or other molecules in the solution, because there is a slight change in pH and your protein unfolds, or because there's a slight change in temperature and as a result your protein denatures, and it could be so many different factors. The total protein decreases because the objective is to remove as much unwanted or non-specific proteins as possible. Right? Cells may have various different proteins, and once you purify through all 5 steps, what is left of your pure protein is much smaller than the total protein. But that is your objective, is to purify your protein. In a successful step, the loss of non-specific protein is much greater than the loss of activity. Therefore, specific activity increases even as total activity falls, as you can see. This is the reason why specific activity is the one that needs to be considered. Protein sequencing is a method that reveals the identity of a protein. Now, why is protein sequencing important? That is because protein sequences are almost unique. And after purifying your protein, if you need to identify and make sure that the protein that you have purified is indeed the protein that you want, a method of choice is protein sequencing. Now, this can be considered as a method similar to some analytical methods that you may perform when you purify a small molecule or an organic compound, right? Now, it's very difficult to compare these two. The analogy is extremely difficult because protein sequencing is an extremely long process. Now, many proteins will have slight variations due to mutations or familial patterns. Remember, protein sequences is mainly amino acids. Now, these amino acid sequences can vary depending on what protein you're talking about, right? And it can also vary in the same protein. And that is where mutations comes into effect. We can determine overall amino acid composition by hydrolyzing the protein and counting each amino acid. That's one method. You can determine protein sequence using the original method that was developed by Sanger. The scientist's name was Fred Sanger, who discovered this method in 1950. And this Sanger sequencing uses proteins to identify the N-terminal residue, the first amino acid residue of a protein. Based on this, they used to identify the protein. Improved methods involves Edman degradation, which can effectively identify up to about 50 amino acids. Now, these two methods are not commonly used these days because much improved methods have already come into existence, such as mass spectrometry. This is one of the most common method that you would turn to if you want to sequence your protein. Now, the requirement for sequencing a protein is not always the choice of a scientist. Sometimes one can also analyze the DNA sequence from which a protein is translated, right? There are lots of pluses and minuses in just analyzing DNA sequence and making sure that it is indeed the sequence that can get translated into a protein sequence. And those we will talk about later. But for the next couple of slides, I would like to talk about Edman degradation and some mass spectrometric techniques. The reaction shown in this slide is what is called as Edman degradation. Now, in this procedure, the protein is first denatured, wherein it loses its three-dimensional structure or a tertiary structure and becomes just a polypeptide. And then the pH is increased and the polypeptide is labeled using phenyl isothiocyanate, this compound, which reacts with the N-terminus of the peptide. And after reacting with the N-terminus, you get a phenyl thiocarbomoyl derivative. Now, this phenyl carbomoyl adduct or PTC adduct is then cleaved in a step carried out in anhydrous trifluoroacetic acid. And when you add trifluoroacetic acid, the pH drops. And what happens is that there's a rearrangement of these atoms. The lone pair on this specific nitrogen moves in, and this nucleophilic sulfur attacks the first carbonyl of this peptide and pushes out the rest of the polypeptide, thereby forming this anilino-thiazolinone derivative of this amino acid derivative. Now, if you look at this, the R prime is the first amino acid in this polypeptide. And then, upon further exposure to low pH, this anilino-thiazolinone derivative gets converted to phenyl thiohydantoin derivative of this amino acid. Right. Now, this derivatized amino acid is extracted with organic solvents, and what it is used, it is then identified. So the use of sequential reactions carried out under the first basic and then the acidic conditions provides a means of controlling the entire process. Each reaction with the amino-terminal amino acid can go essentially to completion without affecting any of the other polypeptide bonds in the peptide. This process, the entire process, is repeated, typically as many as 40 sequential amino acids, and are identified. Now, it is important to know that this is an automated procedure. So, this is just a basic idea as to how Edman degradation is carried out. Edman degradation may work for smaller proteins, but what about larger ones? How do we sequence large proteins? Typically, we use enzymes called proteases that cleave proteins and to digest the protein into smaller fragments. So if you have a larger protein, you need to first make sure to digest them into smaller fragments using enzymes called as proteases, and then apply Edman degradation on these small peptides, or use mass spectrometry to analyze each fragment. You have to repeat this process with a different protease to arrange the fragments. So each protease has got a signature, and they'll only cleave between specific amino acid residues. So what are those signatures, and why are they important? The table shown in this slide gives you a description of some of the common proteases that are used in Edman degradation or mass spectrometric analysis. For example, trypsin from bovine pancreas is one of the commonly used protease in these sequencing methods. Some of the other proteases include submaxillary protease. This is from mouse. Chymotrypsin, again from bovine pancreas, and the aspartate N protease or pepsin. Now, shown here are the cleavage points, which means that these proteases would cleave before or after the residues that are shown here. In brackets, it's written C. This means that these proteases cleave at the C terminus of these amino acids. Let's look at an example. Now, if you look at the peptide sequence shown below, right, and if you use trypsin to cleave this peptide, trypsin would cleave either lysine or arginine at the C terminus. So if you go through this peptide, MFR, R is an arginine, so it will cleave between R and T because this is where the C terminus of arginine is, right? And if you go through the peptide sequence, you find lysine. C terminus of lysine is between lysine and arginine, so it can cleave here as well as here, or it can just cleave after here depending on how trypsin works. There are some trypsin that only works on lysines and not arginines. They've been engineered to do so. Now, let's look at another example. If you were to look at ASP-N protease, it says ASP blue and in the brackets or in parentheses N, right? This means that this protease cleaves a peptide at the N terminus of aspartate or glutamate, not at the C. So if you go through this peptide again, MFRTVYE, E is glutamate, right? The N terminus of E is Y. So between Y and E is where the ASP-N protease cleaves. Again, going through this sequence, KRMSCWD, D is aspartate, right? On the N terminus, that is a tryptophan, W. So it cleaves between W and D. So these proteases are used in tandem or separately to cleave a large protein and to get individual peptides and identify these fragments. Mass spectrometric techniques can provide a much accurate measure of the molecular weight of a protein, in addition to the sequence of a protein. Remember that. What happens in a mass spectrometric technique, or what does a mass spectrometer do? Molecules, or analytes as they are called, and in this case protein molecules are first ionized in vacuum, and the mass to charge ratio of the newly charged molecules, newly charged because they are ionized, right? And these charged molecules are analyzed by an electric and or a magnetic field. So what you're measuring in a mass spectrometer is the mass over the charge ratio. And that tells you, gives you the mass of a specific molecule. In a technique called MALDI, or which is the abbreviation for Matrix Assisted Laser Desorption Ionization Mass Spectrometry, or MALDI as it's called in short, MALDI MS. This is a very robust technique, and it uses laser to ionize protein samples on a matrix and analyzes the molecular weight of the whole protein. That's the key word here. MALDI MS can determine the whole mass of a protein. For example, if your protein's molecular weight is 50 kilodaltons or 50,000 daltons, MALDI is a useful technique to identify. However, MALDI cannot determine various different charge states. To identify different charge states of a protein, you go to another technique called ESI MS, or Electrospray Ionization Mass Spectrometry. This is a very interesting technique. ESI MS is also used for small molecules and peptides. It adds charge to a protein or peptide samples and can be used to determine the mass of whole proteins or peptides. Typically, one uses this technique for identifying the sequence of a protein. MALDI is typically used to identify the whole molecular weight of a protein. The way ESI MS is performed is by taking a protein of your choice and digesting it using protease of your choice, and then determining the masses of the peptides that are being digested. And that will tell you what the original sequence of the protein is. This is a very brief explanation as to what electrospray ionization mass spectrometry is, right? This is the basic. A protein solution, as you can see, is dispersed into highly charged droplets by passage through a needle under the influence of a high voltage electric field. The droplet evaporate, and the ions, these are the ions with added protons in this case, enter the mass spectrometer for M/Z measurement. And that's exactly what happens. The output on the right hand side is a spectrum that is generated, and it has a family of peaks, with each successive peak from right to left corresponding to a charged species with both mass and charge increased by 1. Now remember, proteins have got various different side chains. It has an N-terminal amine and a C-terminal carboxylate, and also in addition to that it has various different side chains, right, depending on what amino acid it has in its sequence. So those amino acids with an ionizable side chains can have various charges. For example, lysine can have a positive charge, aspartate can have a negative charge. So the charged state of a protein can be determined using this technique. It's a very, very useful technique. How are proteins synthesized? Cells make proteins with ribosomes. Ribosomes are the essential part of a cell which makes proteins. And in doing so, it uses an mRNA template to synthesize proteins in a stepwise manner. We as scientists can make use of synthetic biology techniques and genetically manipulate cells into making proteins of our choice. Short peptides, however, are often synthesized chemically, right? The synthesis of short peptides in laboratory is performed using a technique called solid phase chemistry. This involves a solid phase resin that attaches the C terminus of the peptide and the N terminus keeps growing. Peptide synthesis in the laboratory progresses from the C terminus to the N terminus, which means that the C terminus is synthesized first and then the N terminus. This is in contrary to how peptides or proteins are synthesized in cells. In cells, the N terminus is first synthesized, followed by the C terminus. In the laboratory, each free amino acid is chemically protected on the N terminus while the growing peptide chain is free to react. This slide describes as to how peptides are chemically synthesized on an insoluble polymer support, which is called as a solid support. Reactions from 1 to 4, which are shown here, are necessary for the formation of each peptide bond. The FMOG group, as it is called, this is shaded in blue, prevents unwanted reaction at the alpha amino terminus of the growing peptide chain. Chemical reactions proceeds from the carboxy terminus, which is attached to a solid support as shown here, and the growing peptide chain is via the N terminus. Now, you do not have to remember each and every reagent that is being used in this. It is important to remember that the laboratory peptide synthesis happens via solid support, and it starts from C terminus, goes to the N terminus, which is in contrary to peptide synthesis or protein synthesis in cells. Protein sequences help elucidate the history of life on Earth. Molecular evolution focuses on changes in protein sequence over evolutionary timescales. Proteins with similar sequence or structure or function are grouped into families. Examining differences in protein sequence between organisms gives us a clue on key evolutionary steps. This slide gives a classic example as to how one compare protein sequences. And this is displayed by showing the sequence of the protein acidic ribosomal protein P0 or L10E. And this protein is present in almost all organisms, including human beings. And as you can see, the protein sequence starts from 1 and it goes to 76 in this case. However, it doesn't end there. So this is just one example as to how you compare protein sequences. And as you can see, a classic example, K is conserved in almost all organisms that is shown in this slide, right? So these are conserved residues. Lysine is a conserved residue. That's what the terminology is. Knowledge of the sequence of amino acids can offer insight into the three-dimensional structure of proteins as well. In addition to it, it can also give some information on the function of these proteins. Exactly how the amino acid sequence determines three-dimensional structure is not understood in detail. However, there are lots of studies that are currently under progress which is trying to figure out as to how one can determine with accuracy the three-dimensional structure given a sequence of a protein. It is also extremely difficult to uncover or predict the function of a protein from its sequence. We can, however, compare and contrast various protein sequences from different organisms, and we can come up with hypotheses as to how proteins may have evolved from its ancestors. And that is one of the primary uses of comparing protein sequences. Conserved sequences are those that are the same throughout evolution. Usually means they are important for function. Conserved sequences can be either identical or similar. Now, let's look at an example between E. coli, which is a gram-negative bacterium, and B. subtilis, which is a gram-positive bacterium, right? Now, the sequence shown here is for a protein named HSP70. And all those sequences that are shaded in the sequence alignment are identical, right? As you can see, T, T, I, Y, Y, D, D, and so on. Some of the sequences that are not shaded are similar. For example, V and L, right? V is valine, L is leucine, right? I and V, isoleucine and valine. L and I, leucine and isoleucine, right? Now, these are similar because they are hydrophobic and share similar properties, and hence these have similar properties and are also conserved. So that's the example for conservation. Another property is signature sequences, right? Signature sequences are those unique to a group of organisms. And in this case, the signature sequence that is shown here in this box is a 12 residue insertion near the amino terminus of the sequence that is shown here. Now, the signature sequence, as you can see, have identity. It has conservation, is identity, as well as some of these amino acids are similar, right? V, I, right? This is similar. H, K, R, that's another classic example. H is histidine, K is lysine, R is arginine, positively charged amino acids, right? The variation in the signature sequence reflects the significant evolutionary divergence that has occurred at this site since it appeared in a common ancestor of both groups. The example is archaea versus eukaryotes. And as you could see, how eukaryotes might have evolved from archaea by looking at these sequences. And so, protein sequences give a pretty good indication as to how proteins may have evolved, and hence also provides a way to elucidate the history of life on Earth.